Reverse aging. For a long time, aging has been conceived as a natural process of life, and we do not have any control for those processes. Recently, rather than the natural process, aging is considered as a disease. And WHO, World Health Organization, actually gave the disease code for aging. So MG2A is designated to aging-associated decline in intrinsic capacity. Means aging is a disease that implies that there should be a way to treat this disease. The first approach is cellular reprogramming. By adding drugs, if you can change the older cells into younger cells, then this can be one possible way to treat the aging as a disease. In the cellular level, the adult cell or aged cell already has been demonstrated that they can be converted into embryonic stem cell-like cells. This kind of a very young stage of cells, this artificial cell is called as iPS, induced pluripotent stem cell. While this is possible, the one possible problem is iPS can generate the teratoma, benign tumors, or it can go into aberrant differentiation. So to avoid this kind of a problem, one possible solution is converting the aged cell only into the young cell by partial reprogramming. Then it can avoid the side effect of iPS. Then this can be one possible solution to treat aging problem. The reprogramming technique has been applied to restoration of mouse vision. In aged mouse, when the nervous system is crushed, and then the nerve cells died and then fail to recover. So it may lack of the adult stem cells. Here, Yamanaka factor can restore those stem cells, but the original Yamanaka factor has 4 factors, but the c-MYC has a potential oncogenic the dangerous possibility. So excluding c-MYC, 3 factors, OSK has been treated, and then crushing the optic nerve and monitored the recovery of those neuron cells. Very interestingly, the neurons are fully recovered. That means resupplying those stem cells can help to solve the aging-related problem. Yamanaka factor treatment has successfully demonstrated that we can change the aged cell into young cell, but this has been demonstrated only in mouse. This kind of a genetic modification may not be easy to be applied to human being. So later, now it is demonstrated that not the genes, small molecule drugs also can change aged cell into young cell. Then this may have better potential to be clinical applications. The second approach is suppressing SASP. Senescent cells secrete the cytokines, and this is called as senescence-associated secretory phenotype, abbreviated as SASP. And these cytokines induce inflammation nearby, and the senescence symptom is spread out. So here, if you add drugs to suppress SASP, then this can be called as a xenomorphics. Here the morphic means the shape or phenotype. So even though we cannot change the cells into younger cell, at least if we can suppress the bad effect of senescent cells, it can improve the senescent phenotypes. In the cellular level, when the insulin or insulin-like growth factor bind the receptor, then it can activate mTOR and eventually activate SASP-inducing genes. Then SASP can happen and cytokine can be released. For regulation of this process, there is a nutrient sensing system, AMPK. AMPK is usually activated when the cell feels the low nutrient conditions. Then it can suppress the mTOR pathways, and then it can suppress the SASP-inducing genes. AMPK also can activate the sirtuin, and sirtuin suppress the SASP-inducing genes. So left side, this is a growth signaling. Growth signaling makes SASP. And then right side here, this system is a nutrient sensing system. When we have a low nutrient conditions, it suppresses SASP. Now, if you look at those regulators, rapamycin is an inhibitor for mTOR, then rapamycin can suppress the SASP. Metformin is an activator for AMPK, then it can also suppress the SASP. Sirtuin's cofactor is a NAD+, and NMN is a precursor of a NAD, so addition of a NMN also can suppress SASP, the phenotype. So one way is suppressing the activation signal, growth signal, and the other one is the activating the nutrient sensing signal. Both cases can contribute to xenomorphic effect. If the senescent cells can stop the SASP by xenomorphic treatment, that would be good. But if it is too late, cells are too old, then even the xenomorphic approach does not work. Then the only way may be to remove those senescent cells. So in this case, we call it xenolytic approach. Lytic means destroying. So xenolytic means selectively kill senescent cells. Cells have intrinsic apoptotic pathways. Apoptotic pathway is a suicidal signaling. That happens when the intrinsic stress goes up too much. If that happens, then Bex and Beck can make the oligomerization, and then it can generate the rupture of a mitochondrial outer membrane that can spread out the ROS and kill the cells. Now BCL2 is a protecting cell by anti-apoptotic effect, and BID is a pro-apoptotic proteins. So there are several different kinds of anti and pro-apoptotic proteins exist. In senescent cells, the BCL2 protein is overexpressed, so to protect the cells from apoptosis. Here, if we add the navitoclax molecule, which is an inhibitor for BCL2, then senescent cells can be selectively induced apoptosis, and they can die. So navitoclax is a representative, the xenolytic reagent. To summarize the reverse aging strategies, the first one is the reprogramming. So change the older cell into younger cell, but not too young. If the cells are in embryonic stem cell stage or iPS stage, it can form teratoma. Second approach is suppress aging signaling or SASP. So at least the senescent cells cannot do the bad things to neighboring cells. So this is called as a xenomorphic approach. The third approach, if the cells are too old and too late to fix, then the only way is to remove these cells. So xenolytic approach. So these three are the general approach for reverse aging.